Crick referred to the tRNA as an adapter molecule. Let's dive in and figure out why he referred to it as so. Well, first, Merriam-Webster defines an adapter as a device for connecting two parts of an apparatus or an attachment for an adapting apparatus for uses not originally intended. So how does this relate to the function of tRNA, you may ask? Well, let's first look at the functions of tRNA in translation. tRNA acts as the link between the coding sequence of nucleotides in an mRNA and an amino acid sequence of a polypeptide chain during translation. During protein synthesis, each tRNA brings an anticodon to the corresponding codon in the mRNA sequence attached to the ribosome. The tRNA anticodon helps code for a specific amino acid to create the polypeptide chain. Therefore, it acts as an adapter between the mRNA and the amino acid, aiding in the translation process. So here we have our tRNA with the amino acid. It'll go to the corresponding codon, and it has the anticodon, and it'll eventually make a polypeptide chain of amino acids. Let's look at a technological analogy. So here we have our phone representing the ribosome, and it has the mRNA coding sequence on it. So the two headphones are going to be the amino acids, and the new adapter on the new headphone is the tRNA. So when the new iPhone came out, there was no way for this to get into the phone, right? So this isn't going to work because it doesn't have the adapter tRNA. So as the tRNA acts as an adapter, it can link the anticodon and the codon sequence. Now, what are some instances that the tRNA may not act as an adapter that conforms to the genetic code? Well, the third base in the codon and anticodon translation process pairs weaker than the first two bases, and sometimes the pairing differs from the common Watson-Crick base pairing of adenine to uracil and guanine to cytosine. This can make it so the same tRNA can code with multiple different mRNA codons. As for the effects in the cell, if a wobble occurs and a different amino acid is added to the polypeptide chain, a different protein could be produced, or more than one sequence can code for the same amino acid as well. This all just alters what polypeptide chain is then produced in the cell.